I'm James McClaren. I'm going to talk about some of the terms that Dr. Schierbaum used in her presentation. She spoke of HSP as being a differential diagnosis of CP, cerebral palsy. That means a differential diagnosis is like a list of possible diseases that fits the symptoms that a person has. That it's kind of a, oh, it could be this, this, and this, and you kind of try to narrow it down. So she's saying that people that maybe physicians think they may have CP, it's possible that they have HSP. That's something that they should look at. Um, she talks about diagnostic yield. That is you know, the number of uh, cases that are successfully diagnosed. Like we know what causes that disease, what percent uh, do we get a successful diagnosis? That's the diagnostic yield. Um, a CNV is a copy number variation. It's a type of genetic mutation where, like many genes have repeated segments, and it's a difference in the number of repeats from one person's genome to another person's genome. And the first patient she was talking about, because of the diagnosis that they made, that particular gene, there's a history sort of cardiomyopathy, which is basically a something that affects the heart muscle, like a heart disease problem. So they were saying that she should be uh, treated with probably one of the things they might treat her with is CoQ10 or coenzyme Q10, which is something you might have heard of. It's an antioxidant, but it's used uh, for sort of to try to lessen the possibility of congestive heart failure in those patients. So it's something she should be having. Optic nerve hypoplasia is an eye a loss of sight condition where the optic nerve, the nerve that goes from your eyeball to your brain, is underdeveloped. She mentions dysmorphic features. A dysmorphic feature is a difference in body um, sort of type or structure from the norm. Something like a variation from the norm that is sort of indicative of a disease. Um, she mentions a splice site mutation. So, um, in genes that make proteins, that are the code that makes a protein, there are often these little segments of DNA called introns that are chopped out before that gene goes on to be used as a pattern to make a protein molecule. Um, so the splice site is the specific pattern of DNA that causes that intron, that piece that gets chopped out. That's the enzyme, the little machine that comes along and chops that piece of DNA out. Um, it recognizes this specific pattern of on the DNA, and that tells it, oh, latch on here and start cutting. So if there's an error in that splice site, that can cause really big problems with your protein molecule, because that intron that's supposed to get chopped out maybe doesn't, or it gets chopped at the wrong spot, and you get some of that stuff that's supposed to be chopped out being used as a pattern for your protein molecule, which means your protein molecule is not correct anymore. So it's quite a severe potential malfunction in many cases, or mutation in many cases. And that's about all I wanted to mention from Dr. Schierbaum's presentation. Thanks for listening. Hi, I'm James McClaren, and I'll talk a little bit about Dr. Zuchner's presentation. So he talks a lot about gene sequencing, and I wanted to sort of give you an idea of how that works. Um, the genes in your cells, the DNA in your cells, consists of a long string, thousands and thousands of these things called nucleotides, and each nucleotide has a thing called a base on it. All we really care about for our discussion here is there's four different kinds of bases, and we're just going to represent them with letters. It's the first name letter of the name of the thing, and they're A, C, G, and T. Okay, So what your DNA consists of is essentially a language of 4 letters, okay, that goes on and on and on. You actually have 46 little books of these things that consist of this whole string of letters. You have 46 genes in your DNA, in your genotype, we call it. Of those 46, you get 23 from your mother and 23 from your father, and they're all essentially duplicates. So there's really only 23 sort of unique ones. Anyway, how do you the process of reading a genome? I just wanted to kind of talk a little bit about that. It's not as simple as you just sort of look under a microscope. Oh, CAGTGCA. You can't do that. It's quite complex. It's too small. You can't see it like that. So there's essentially two means of reading the DNA. One's called short read sequencing, and the other one's called long read sequencing. So in short read sequencing, I was trying to think up an example of how this would work. Basically, you can only look at the pattern of short little pieces of the DNA at a time, 50 to 300 of these things called bases, these letters. You can only read little snippets of it. So think of it, here's my little example. Let's say you had the script of a movie, okay? And the movie scripts, the production process continues. They get rewritten and modified, but there are certain similarities, right? From one version of the script to the next, okay? So your genome's kind of like that. Like your genome, your movie script is similar to your mother's and your father's, but not the same because it's a mixture of the two. But more than that, every human being in our cells kind of do the same things, and the genome is very similar from any one human being to another with slight differences. Okay? So think of that as the script for this particular movie has many variations. And let's say somebody, the latest rewrite of the script, somebody accidentally shoved it in a shredder. Okay? Oops. But you want to be able to read that. So one way to do that would be take all those little shredded pieces and compare it to an intact script for your movie. As let's say in the original script there was a sentence, the quick brown fox jumped over the lazy dog, and later that was changed to the quick brown fox jumped over the energetic dog. I don't know, it's a stupid example, but it's trying to explain. So that little piece of shredded paper that says quick brown fox jumped over the energetic dog, you could align it with the original script, like the parts that are the same, you could align, and then you see, oh, there's a difference. Okay, so that's basically how gene sequencing works, believe it or not. You take these little short pieces and you align them with a kind of an original, like your original genome. That's why the original production of the human genome is very important, because it's used to sort of figure this out. Now, one big problem with this short read sequencing process is there are a lot of places in the genome where you get what are called repeats, where it just has the same sequence of letters like CAG over and over and over and over for hundreds and hundreds of units. So if that's the case, it gets really hard to figure out where that little shredded piece fit into the original script, because it's just like, how do you align it? Because it's just a whole bunch of repeats. How do you know where it starts and where it stops? Like if your little shredded pieces go CAG 50 times, does that line up when, and the original script has CAG 200 times in a row? Well, where does that little piece line up? Is it right at the very beginning of that, or the very end, or somewhere in the middle? This is really hard to figure out. Okay? So that's where long read sequencing comes in. You can read longer chunks of DNA. You don't have to break it up into this little 50 unit little shredded pieces, and it's much easier to figure out where it goes when you could do 3,000 letters in a row instead of 300. It makes your life much easier. um, I hope that little example made some bit of sense, but to sort of understand that genetic sequencing is not simple. It's a complex process, and it's difficult. And the other thing that makes it difficult is a lot of the DNA. Like we often talk about it, the DNA, the genes coding for proteins, making these proteins, these little machines inside the cells. A lot of it doesn't do that. Only about 2% of your DNA actually codes for proteins. The rest of it is what we call non-coding DNA. One time they used to call it junk DNA because they thought it was just sort of there and didn't really do anything. But that seems to not be the case at all. It's actually very important. It seems to have a role to play in sort of the kind of physical structure of the DNA and how it is transcribed, like a process of causing it to different parts of the genes to activate or deactivate. So for this reason, it's looks like it's important to be able to read these non-coding parts of the DNA in order to learn more about genetic diseases, basically. Um, he mentioned introns and extrons. I think I've talked about those before. In a sequence of DNA in your cell and a gene, before it's converted, like coded into a protein molecule, they're actually pieces of those, uh, the sequences of these letters A, C, G, T that are chopped out. It's not 100% understood why that happens, but these are called the introns. They're chopped out before that thing goes on to make a protein. So again, they don't seem to do anything, but Dr. Zuchner's research seems to indicate that in some cases the pattern of these things called introns changing has a role to play in whether the disease is expressed or not. The error or the mutation in the gene causes a phenotype issue, an issue for the actual mechanisms of the cell, I guess you could say. Um, there's a slide in there where you talked about meiotic instability. Meiosis, Meiosis, is the process that makes the like the human sex cells, the egg cells and the sperm cells. Each of those cells has half the normal amount of genes. So they each have 23 instead of 46. And then those two things come together to make a new individual, a new person. A new person's born with half the genes from the mother and half from the father. Uh, what he's talking about, it seems to be a very kind of unstable process, and there's a lot of this repetition stuff occurring, or like a gain of the repeated sections of the DNA or a loss of the repeated sections of the DNA in the process of making a baby. Is basically what's going on there. And he mentions modifier genes. So yeah, it seems that um, like we know with HSP, um, some people that seem to have the same mutation get like express the disease early on in life, and some of them much later, um, and uh, and there's a lot of variability in that, in terms of when that onset is. It's not really well understood at all why that happens, and Dr. Zuchner thinks that there may be modifier genes, the presence or absence of other genes types, like mutations other than the specific, SPG4, SPG7, or 11, or whatever it is, um, might be responsible for the time of onset of that disease. So if we could study those and know more about those, we might be able to control whether the person kind of expresses the disease or not. And I think that's all about it. Wanted to add to Dr. Zuchner's presentation. Thank you. Hi, I'm H.McClaren, and I'm going to talk a little bit about Dr. Calame's presentation. So he talks about genetic sequencing and HSP that's undiagnosed genetically is mostly what he's talking about. Um, so there's a couple terms, genome and exome. So um, your genome is all of your genes in the DNA molecules in your cells. Um, as we already talked about in previous little discussions, a lot of the DNA does not code for proteins. Like we think of the DNA as being a code for these proteins, these little machines that do work inside the cell. A lot of it doesn't. A lot of it consists of one of three things: introns, which are pieces of DNA in the middle of a coding sequence that get kind of chopped out when that piece of DNA is then used to code for a protein molecule. There's chunks that get taken out. There's non-coding DNA, which is just seems to be sometimes called pseudogenes. It just doesn't seem to code for anything. Not really sure about that. Why it's there. And then there's structural DNA, which has to do with basically just the physical structure of the DNA molecule, the making it able to duplicate itself for cell reproduction and stuff like that. Doesn't code for proteins. Anyway, so a lot of what Dr. Calame is talking about is some of these bits of DNA may be responsible for the onset of the condition. So maybe we need to look at that DNA as well as what's called the exome. The exome is the part of the DNA that actually codes for proteins. And usually when a person goes for DNA testing, they only look at the exome, and typically they only look at certain parts of it. So if someone is diagnosed with HSP, they will look at certain parts of the DNA exome that tend to contain the genes that are known to cause HSP. So what he's saying is you need to go beyond that in testing for a bunch of different reasons. So he talks about three different sort of scenarios. So he talks about HSP mimics. So that would be essentially another disease, a different disease that in the sort of the range or spectrum of symptoms of that disease. In some cases, it can look like HSP. So essentially, it's some other gene that's causing this. It just looks like HSP. Phenotype expansion is kind of the same thing. It's an extreme range of symptoms of some other disease that we're seeing. And a novel association is a gene that we haven't known previously that causes HSP. I mentioned exome sequencing. So whole exome sequencing is where you look at all of the exome, all of the parts of the DNA that are responsible for making proteins. So he's suggesting that is a useful approach to diagnosing diseases like HSP, because you don't really know for sure where that genetic mutation is. It might be somewhere where you're not expecting it. He mentions the patient with hemiplegia. Hemiplegia would be a paralysis of half of the body. I just wanted to explain, he talks about pumps and cation leaks and stuff like that. So every cell in your body is surrounded by a plasma membrane that separates it from the other cells and the stuff in between the cells. And that membrane keeps it's like the skin of the cell, I guess. It keeps the contents of the cell in it and things that aren't supposed to be out. So there are various little, essentially proteins coded by DNA, embedded in that membrane that are responsible for removing things that need to be taken out of the cell and bringing in things that need to come into the cell. In neurons, there are a couple of different pumping mechanisms that are responsible for the nerve signals that are triggered across the cell. Basically, when a neuron fires, as we say, it sends a little electrical pulse through its membrane. And breakdown of the function of the pumps that's responsible for that could be a cause of a lot of different neurological diseases, because the neuron, the cell, is not going to send nerve signals properly if that occurs. Untargeted genome sequencing, he mentions, that is, again, where we look at the whole genome and we're not looking at specific parts of it, expecting a mutant gene to be in a particular area, because that's where it is in most other variants. It's sort of looking at the whole thing. Full genome sequencing compared to exome sequencing is very expensive in comparison, so that's not something that's done for genetic testing very often. And he talks about another sort of membrane pump called SLC7A1, and he talks about arginine and lysine and ornithine. So those are amino acids. Amino acids are the building blocks of protein molecules. So in some cases, the cell makes these little building blocks itself. In other cases, they're obtained from your food, from your diet, and they have to be brought into the cell. So that's what that pump would do, be pump those things into the cell. They're the bricks that build the protein molecules that do the things that the cell does. And if you don't have those bricks available because they can't get through the membrane, through the outer surface of the cell, then your cell's not going to work very well. And the urea cycle, that is a mechanism inside the cell that removes kind of waste products from the metabolism, from the energy processes of the cell. Specifically, in this case, it's nitrogen atoms, which are a byproduct of that process. I think that's all I wanted to talk about from Dr. Calame's presentation. Thanks a lot. Hi, my name's H.McClaren, and I'm going to talk about Dr. Qiang's presentation. To begin with, he talks about organoids and stem cells and all kinds of different cells. So to understand what he's talking about, you have to remember, think about a person starts out as a single cell, right? So a human egg cell and a sperm cell come together, and that forms a thing called a zygote. And a zygote is an example of a stem cell. A stem cell is a cell that can turn into other types of cells. So it could turn into a blood cell or a neuron or a skin cell or whatever the case may be. So that's what a human stem cell is. It's a cell that can grow and duplicate itself through a process called mitosis, which makes an identical, two identical cells. It just doubles and doubles and doubles again and again. And that's how a person is made. They start out as a single cell. It turns into two cells, and each of those divides again and again and again until it gets to this little blob, and then they start to differentiate into the different types of cells. He mentions somatic cells. You hear that term quite a lot. Somatic cell is just a body cell, any differentiated body cell. And then he gets into iPSCs or HIPSCs. HIPSC stands for Human Induced Pluripotent Stem Cell. What that is a fairly new technology where you can take a normal human cell, usually it's a skin cell or a blood cell, certain types of blood cells, And you can sort of treat it in a way that causes it to undifferentiate, to become a stem cell that can then go on and turn into other kinds of cells. It's a very powerful research technique. And what you get, you grow these cells in a lab in a culture, in like a petri dish, basically, is similar to that. And you get a whole bunch of these undifferentiated stem cells. And then a really cool thing you can do with those is turn them into things called organoids. So you can cause those stem cells to turn into various different types of cells. I don't know the details of the technology for how they do that. It's pretty complex, but we won't get into that here anyway. And what you can do with those differentiated cells is you can form these little things called organoids. Think like an organ of the body, like your brain or your liver or your heart or whatnot, is formed of a bunch of different types of cells. Like your brain is not just neurons, there's other little helper cells in there that make it work. So that's what an organoid is. It's a sort of an artificially produced little blob of cells that are not just one type of cell, but a bunch of different types. So in this particular example, we're talking about brain organoids, or mini brains, they sometimes call them. They're, a millimeter or two across would be like an eighth of an inch, something like that. They're quite small. They're not little human brains floating around in a dish. They're sort of groups of neurons and connected cells. So they're very useful for research purposes. You can grow a whole bunch of them, and you can do stuff to them. You can add drugs to them and see what the effect is. Pretty quickly, sort of leads to new research. In the process of making the organoids that Dr. Qiang was talking about, he mentions a thing called CRISPR. That is an acronym. The letters are CRISPR. It is a technique in which you can essentially take a piece of DNA out of a living cell and replace it with a different piece of DNA. And it can be very specific. It's a certain piece. So you can basically, what he is doing in his research is taking a normal, like he's taking these iPSC cells, these induced stem cells, starting out from a person who has no mutations, like has no diseases, has a normal genome, and they're taking a specific piece of mutated DNA in that SPG4 gene and sticking it in there using that CRISPR technique. So the purpose behind that is essentially what we call in science a control mechanism. The only thing that's different about that cell is that mutated specific gene. Everything else is sort of the same. So you're starting out with all those cells are the same except for that one little thing. You hear the words genotype and phenotype a lot. Genotype is all of your genes collectively, all of them. Phenotype is your characteristics. So we often hear, everything's determined by your genes. Well, how specifically? Phenotype is what the organism or the cell does, like maybe it expresses a particular disease trait. Genotype is just the information contained in the cell that makes all of the parts of the cell, basically. So we want to understand how those things are connected. The doctor mentions a couple of types of gene mutations. He talks about a truncated mutation and a missense mutation. So genes produce these things called proteins, which are like little machines inside the cell, which consist of long chains of these molecules called amino acids. There's about 20 different amino acids. A truncated mutation would be where that resulting protein molecule is shortened. There's like a whole piece of it missing, like a long section of it. A missense mutation is where one or more of those amino acids in that protein are changed. They're not what they're supposed to be in the normal gene. CTIP2, GFAP, SMI32, those are all different enzymes, different protein molecules that are present in different types of cells, and they're used in his research as markers. So you can basically you take an antibody, which is just a molecule that attaches to a particular protein molecule, and you basically that antibody has like a phosphorylated piece on it that basically it causes it to glow in the dark, and you can measure how much of that is like particular types of cells are present in your culture by causing it to glow in the dark and making it light up. Basically, it's pretty cool. Apoptosis means cell death. Cell death, that sounds bad. Not always. Basically, an organism or a human being is a whole collection of many millions of cells, and some of those cells don't work properly, or they get kind of used up in the process. Like think things like skin cells, they get kind of worn out. Cells along your digestive tract get kind of worn out with time, and they kind of have to die once they've done their job. And so apoptosis is not necessarily a bad thing, but sometimes obviously it is. He talks near the end about astrocytes. Astrocytes are a type of cell that's present in your brain that kind of supports the neuron cells and helps to basically get energy and food and nutrients to those cells. It's a part of your brain. Like I said, your brain is not just neurons. There's other cells in there kind of in the mix that help out. And he talks a lot in the end about HDAC6. Again, HDAC6 is a particular protein that's present in cells. In people who have the mutation, the SPG4 mutation he's talking about, they seem to have too much of it. What this protein does is it's connected a lot with one of the things it does is transcription, which is the process of making a protein from the DNA, and it inhibits transcription. It stops this from happening. It's one of the things that it can do. So it seems that if you inhibit it, if you try to reduce the amount of this HDAC6 or reduce its activity, it seems to help those cells with the mutation. So it's a promising target for therapy because there are drugs that you can use that will inhibit this HDAC6 molecule. And that's about all I wanted to say about Dr. Qiang's presentation. Hi, my name is H.McClaren, and I'm going to talk to some of the details of what Dr. Piermarini spoke of in her presentation at the conference. So she talks about this SPAST C448Y mouse model. What the heck is that? It's a mouse that has had the mutant SPG4 human gene inserted into its genome. It also has the normal mouse genome for the SPAST gene that produces the spastin protein. So it's not exactly the same as a person that has HSP, but it expresses that sort of mutant defective protein in it, and it shows kind of some of those symptoms of this disease. And being the mouse has been altered by gene therapy, and then all of its little kid mice, the pups, will also express this this mutant human gene. So you have a line of mice that you can experiment on, all of which have this sort of defect in them, this mutant gene. Um, so she's talking about a means of gene therapy called AAV, or viral vector gene therapy. So the way a virus works, when you get infected by a virus, the virus attaches itself to a cell. Usually there's a specific cell that it targets, but it attaches itself and it injects sort of its own little DNA molecule into your cell, and that DNA gets incorporated into the DNA in your cell, and then it gets expressed. So what that DNA does is make all the parts of the virus. So what you've done, or when you get infected by a virus, the virus basically sort of takes over the machinery of the cell to make copies of itself. That's normally what happens when you get infected by a virus. So in order to use that for gene therapy, basically you have a sort of a specialized virus that instead of containing the DNA of the virus, it contains the DNA that you want to insert in the cell. So you essentially infect the cell with a virus that contains the normally functioning gene for the thing you want to give to that cell. In this case, it's the normal functioning human spastin protein that you create. Now, in order for this to work in the mouse model, the other thing you have to do is add a like a protein or a virus that basically what it does is it stops the mutant spastin, the gene that you've earlier introduced into that mouse model, from actually functioning. Otherwise, you don't really accomplish anything because you're still making the mutant protein as well as the normal protein. So there's these two steps in this approach. You have this sort of virus thing that adds a thing. I think it's an antisense oligonucleotide is the thing that they use, but it basically prevents the mutant gene from expressing itself, from actually making protein. And then the second thing you do is you add the normal gene to that cell. And that's what happens. So in the study that they've done so far, she talks about P0, P1 mice. That's postnatal 0 to 1 week after the mouse is born. So it's just a little baby mouse that they're working on. And she goes into the results, and they're pretty positive. I think we really need to talk about that. Doesn't mean that it's a cure. A mouse model is a long way from an approved therapy. Mice aren't people. They're the way their cells work is pretty similar, but we don't know if it's going to work in people. There are a lot of steps involved, a lot of research, a lot of money needed. Clinical trials are complex and expensive, and I know patients kind of get concerned of, why do we have to do all these steps? Well, we don't know for sure that it's not going to be harmful in people. And we're talking about neurons in your spinal cord. One thing that's worth noting is once you're born, you don't get any more of them. So if there's a harmful effect for this thing, it could make things worse. Also remember, these are just newborn mice before they've actually kind of had any symptoms of the disease. So as a therapeutic treatment, if it has to be done before you have any symptoms, that's a bit problematic. How would you know what people to use it on? Now then she talks about sort of next steps, and they want to do is use basically the same procedure on mice that have already kind of shown symptoms of the disease of HSP, say mouse model, and combine that with exercise, which is basically putting the mouse on a treadmill, making them walk. That's been shown in cases of like spinal cord injury to be effective treatment. A couple of things she talks about: neuronal plasticity and axonal sprouting. That's basically nerve cells recovering from damage. They get damaged and they start growing new connections to muscle cells and to other nerve cells. Trying to see if that will happen in this particular case. She mentions neurotrophic factors. Those are various different compounds that help neurons to survive and grow and prosper and be, healthy. So what she's talking about there is, in this experiment, they're going to look for these substances after the mouse is examined, after it's biopsied, essentially, to see if those compounds are present, that show that its neurons are recovering from the damage. That's really what we'd like to see, isn't it? The last thing I wanted to mention was a question asked about a deletion mutation. I'm not sure how to get into this in detail. There's all kinds of different types of mutation. Maybe if folks are interested, I could do a little presentation on that. So a deletion mutation is where one specific little, it's called a nucleotide, this little portion of the DNA that codes for a protein is missing. It's one type of mutation that occurs. And there are different ways that can occur. And usually what it means, as the doctor said in her presentation, if one of these deletion mutations occurs, the protein kind of stops at that point where that mutation occurred, and the rest of it doesn't really function properly. So, if your protein's supposed to be this long, it ends up being this long, maybe. Like it's cut off at a certain point, so it's probably not going to function at all where a deletion mutation occurs. And it's just one further thing on mutations. This particular spastin protein, as an earlier presenter was talking about, most of the time when mutations occur in a protein, it actually has no effect on the function of the protein. A lot of it is just structure of the protein, like whether you're a little bit taller or a little bit shorter, or a little bit wider, a little bit narrower, you still can be a person, right? Well, proteins are kind of the same way. If there are only slight changes in their structure, it often causes no effect in how they actually function. But this particular protein, there seems to be many. There's over 200 different, or the SPG4 version in particular, there seems to be over 200 different possible mutations, all of which cause like negative effects. So it seems to be a particular protein that's super sensitive to errors, to mutations. And that's about all I wanted to talk about for Dr. Piermarini's presentation. So thanks for listening. H.McClaren, and I'm going to talk about Dr. Mitsumoto's presentation from the Friday conference, SPF conference. So Dr. Mitsumoto studies PLS, which is primary lateral sclerosis. It's related to, he thinks, ALS, amyotrophic lateral sclerosis. And the doctor discusses the differences between the two diseases in the question. In his presentation, he does a really good job of thoroughly covering that, I think. Couple of things he's talked about. He talked about a startle reflex. So that's when you hear a noise behind you or something, So that's kind of an automatic response, I guess. It doesn't really connect to your brain. It's quite a interesting thing if you want to read up on it. And he was talking about how that is expressed in PLS patients. It seems to be a common characteristic. The main thing he talks about in his presentation is what's called a natural history study. That is a study that looks at the specific symptoms of a particular disease and how it progresses and how you can measure that and measure changes over time. That's really important when you get into trials, clinical trials for a therapeutic treatment, for a treatment that reduces the progress of the disease or improves a patient's condition. In order to do a clinical trial to measure whether a particular drug, for example, is actually having an effect, you have to be able to measure that. You have to have sort of really clear criteria that you can judge whether that particular drug is improving the patient's condition or not. That way you can tell your drug's doing something. So in order to be able to do that, you have to know something about the uh, the progression of the disease, like how it changes with time for patients, on average, like over many patients. Um, so it's really important to have that in place before you have clinical trials really able to proceed at all. Um, so he speaks about ALS FRS. FRS stands for Functional Rating Scale. So that's a specific measurement tool that clinical doctors will use to measure the progress of ALS for a patient. And he was talking about how this is used for PLS patients, but PLS patients, uh, it the disease progresses more slowly than it does for ALS. So the problem is, like, basically what he's saying is you need to have a specific scale for PLS patients, which is going to be slightly different from the ALS FRS, in order to measure change with time. And again, you need that if you're going to do a clinical trial, so that you can measure whether you're having an effect on the patients or not. So that's why it's really important. We spoke about biomarkers. And again, a biomarker is sort of a specific thing you can measure in something like a blood test, for example, that would be an indicator of the disease, or perhaps like a level of a particular protein in a blood sample might be an indicator of the progression of the disease. So that to be able to identify those sorts of things, again, in particular for clinical trials, is really important because you can measure how well the patient's progressing and how they're changing with time. Bulbar symptoms might be a question for some people. Bulbar is related to the mouth and the throat. Why did people who don't have bulbar symptoms are excluded from the PLS natural history study? Because you don't want to include people who actually have HSP in your natural history study of PLS, because there are different diseases, they present differently. Some of the symptoms can be similar. So if you have people with HSP in your study, then you might not get the correct natural history progression of PLS specifically. That's the issue that they have. And typically HSP patients don't have bulbar symptoms, although that's not 100% always the case. He spoke about a couple of different tests that they use in the natural history. The Pataka test measures basically speed of speech. So they measure the time that it takes you to say pataka, pata, or pata, pata, pata over and over again. Sounds a little weird, but it's a way to measure like how long that nerve signal to make that noise takes to get from your brain to your mouth, basically. The finger tapping is another test that's used to measure the speed at which those neurons are passing signals along. So that's something that you can measure change with time relatively easily. And NeuroQOL is QOL is quality of life. So it's like a questionnaire, like a survey. That's a standardized thing that you can use to measure the mental, physical, social effects of the disease on the person, and how that's changing with time. The last thing I wanted to mention, Dr. Mitsumoto is very much of the opinion that diagnosing these types of diseases, both PLS and HSP and other neuromuscular diseases, has to happen faster. Thinks once therapeutic treatments become available, he's firmly of the opinion that the faster those treatments get applied to the person, the more they're going to have the likelihood of improving their quality of life. So that's why he feels this natural history study is really, really important, because it will help doctors to diagnose patients earlier. Particularly, if you had biomarkers available, that would really help to speed up the diagnosis. So that's about all I wanted to say about Dr. Mitsumoto's presentation. Hi, my name is H.McClaren, and I'm going to talk about some of the terminology that Dr. Baas used in his presentation. Now, to do so, I'm going to use a little diagram here. So that's what you're seeing on your screen. This is a little picture of a neuron. So the neuron has a long, sticky outy part, and that's called the axon. These little things that look like pillows here are a thing called myelin sheaths. They're actually come from a different cell that's kind of wrapped around these long axon things. It's basically insulation that protects the signal traveling through this neuron from all the other neurons that are nearby. Now, these little ends sticking out here are what are called the synapses. These little sticky out parts at this end, those little guys, are called dendrites. So the dendrite of one neuron can connect at the synapse to the axon of another neuron. So they many of them can be connected together. And that's how nerve signals travel through your body and between the cells in your brain. The axons can also attach to muscle cells and connect and energize the muscle cells and cause them to contract, and that's basically how your muscles work. So a couple of the things that happen in HSP is the axon part can swell, and that kind of causes the synapses to sort of move backward and lose the connection with the dendrite of the next neuron, and then the nerve signals aren't passing through properly. That's one of the effects that occurs. Now, someone when he talks about it gets into how DNA works. So I'm just going to move this up. We're going to talk a little bit, sort of very briefly, about how DNA functions. So he talked about this ACTG stuff. A DNA molecule, I'm not going to get into it in a great lot of detail. It's a very, very long molecule that consists of many, many little subunits that are attached together. So those subunits, if you want to know, their names are adenines, cytosine, thymine, and guanine, but we can just call them ACTG. They can be in any order. Any one of them will link to another one. And if you know a little bit about DNA, you might know In DNA, there's actually another strand that's attached to this, but we won't get into that for now. We don't want to get too complicated. So each group of 3 of these in succession, and it's going to continue on and on. That's what we call a codon. The word codon kind of sounds like code. That's basically what it is. It's a code for a specific thing called an amino acid. So each group of 3 of these is like a word that represents one of 20 different amino acids. What are amino acids? Those are the things that link together to form protein molecules. And proteins are like the machines of the cell. They're the things that do all the work of, well, basically keeping a cell alive and doing all the things that it does, and making the things that it does. So the little hair follicles in the top of your head make proteins that create hair, and so on. All different cells make all kinds of different stuff. Now, a mutation can occur when, sort of during the process of replication, when the DNA is duplicated to form new cells, mistakes can occur. So maybe during that process, this little guy, this little G here, is mistakenly replaced with a C instead. So what that can do is cause a change in the protein molecule that this thing codes for. So that's what we call a mutation. That's how it happens. These things, it's not a 100% perfect system. It goes wrong sometimes. So that creates mutations. They happen all the time. And when a mutation happens, really one of 3 things will happen. Most of the time, it doesn't make enough difference to the resulting protein that it acts any differently, and it just does its job, whatever it's supposed to do. So a lot of times that's what happens. Sometimes the protein molecule will not do what it's supposed to do. So that's called a loss of function. Loss of function, you think, oh, well, this is not going to work, so that's okay. Not necessarily, because then you can get a defective protein that just kind of builds up in the cell, and that in itself can cause its own problems. And then the third thing that can happen is a gain of function. So a gain of function is when it does something else that it's not supposed to do. And obviously that can be really bad. It can be like toxic, like it can cause damage to the cell in some way. And that seems to be the case in particular with SPG4. That's what happens. Now, a couple other terms he uses are exons and introns. Well, what the heck is that? So if we just extend this and make up another little pattern here. So it turns out like these sequence of codons in the DNA, when they're sort of translated to make a protein molecule, some pieces are actually kind of snipped out. And they don't actually go on to make a protein molecule. So those snipped out pieces are called introns. They're like, and word comes from, they're like intruders in the code that are just chopped out. And the rest that are maintained are called exons. So the exons are the actual code for the protein molecule. And the introns are pieces that are in the DNA molecule, but they don't actually form code for the protein. And you're probably going to ask, why are they there? Good question. It's not 100% known why it works that way, although we think they probably have a lot to do with telling that particular gene when to turn on, like when to actually make that protein and when not to, like controlling it, turning it on and off, basically, has a lot to do with it. So, whoops. Next thing I wanted to talk about are this M1 and M87 proteins that he was talking about. So the M1 protein seems to be associated with microtubules. So microtubules are like, well, tiny tubes. That's what the word means. They're like a skeleton inside the cell. So they're tiny little, again, protein molecules that form a kind of a network in the cell. And they're like a skeleton. They're also like a transport mechanism because there are other proteins that kind of, like little inchworms, they kind of inch their way along these microtubules and they basically carry things back and forth, which is pretty cool. So the M1 protein seems to be associated with zipping those, which, why would you want to chop them up? Basically, to control them, to maintain them, to make them do what they're supposed to do. And the M87 seems to do with membranes. A membrane is, there's an outer membrane on the cell that keeps it separate from the outside. But what we're talking about here is a large kind of structure of inner membranes. They're like, the land inside the cell. They also help to maintain the structure of the cell, and there are also places in which various little things called organelles, little portions of the cell that do different functions, are kind of jammed in there. And their job is to provide a place for various different functions of the cell to occur. That's mainly what they do. And if they're messed with by an incorrect protein, that can definitely cause a lot of problems as well. I think that's everything I wanted to cover. Oh, one other thing that he mentioned are something called ASO, antisense oligonucleotides. So that is a mechanism by which you can basically do gene therapy to turn off a gene, to cause a gene to not function anymore, to not be expressed, to not make its protein. And then one other thing he mentioned was antibodies. So he talked about an M1 antibody. So what that is, that's its own protein molecule. Cells are really tiny, and protein molecules are really tiny. So this M1 is a particular protein molecule inside a cell, and basically we want to be able to measure it and to know if it's how much is there and how much isn't there. So what an antibody does, it's another protein molecule that basically hooks on to a different molecule, to a specific molecule. So that's what it does. So you design an antibody molecule that hooks onto this M1 protein, and the measurement device actually measures the presence of the antibody when you're doing a test, but being hooked onto that protein molecule, measuring the antibody measures the protein, the presence of that M1 protein. So that's how that works. It's also possible that by generating this antibody and giving it to a person, if the problem is a gain of function, in other words, the defective protein molecule is doing something that it's not supposed to do, it's possible that attaching this antibody to it will cause it to not do that anymore. It's kind of like you've tied a big chain around its leg, and it can't walk around in the cell and do whatever nasty thing it is that it's doing. That's kind of a hit and miss proposition. Whether that works or not, we don't know whether that's going to work or not, but that's the idea. So it could actually, it's quite a exciting sort of development. It's possible that these things could have like a therapeutic application. And I think that's pretty much what I wanted to talk about. So that will end this video. Hi, my name is H.McClaren, and I'm going to talk about Natalie Downs' presentation. So she talks about a transcriptome. What the heck is that? Well, I've got to back up a little bit and talk about genes and DNA and all kinds of stuff. So when you're born, you started out as one single cell, right? And the genes in that cell, half of them came from your father, a little thing called a sperm, and the other half came from your mother in an egg. And then that one cell divides and divides and divides over and over and makes all the cells in your body. So you started out with a whole bunch of genes from your father and a whole bunch of genes from your mother. How the heck does that all make all your genes? Well, essentially you get 2 copies of every gene that you need. One from your mother, one from your father. These are called alleles. So for example, there's an allele for your eye color. And there's kind of 2 versions of it. You can have an allele that ends up giving you brown eyes, or an allele that gives you blue eyes. So every allele for every trait that you need for every cell in your body is contained in every cell. So there's a lot of them. There's something like 20,000 different genes in each cell. But a lot of those cells don't need to express those alleles. In other words, so the cells in your skin on the top of your head, generate the proteins that create hair, and some of us more than others. Your neurons in your spinal cord, they don't need to make hairs, right? But they have the genes to make hairs. And I talked about eye color. Well, the cells in your retina, not in your retina, but on your iris, in your eye, whatever, they have to make in my case, blue color, or in many cases, a brown color, to color the iris of your eye. Well, you don't need that in your skin cells, but your skin cells have the allele to do that, right? So how does that work? So there's DNA inside your cell, and there's a DNA gene for every single trait of every single cell in your body. And then to make the proteins, which are the little machines in your cell that actually do stuff, to do the actual work, what happens is the DNA is, we say, transcribed into a slightly different molecule called RNA. And the RNA goes on and acts as the template, the plan to make these proteins that I was talking about. So different cells in your body only express specific genes at a time. They only make RNA for a few genes at a time. So when we talk about a transcriptome, what Natalie's company does is they look at only they basically measure the RNA that the cell has produced, not the DNA. So they're measuring what that cell is actually actively doing in that little petri dish, basically, is where they are. It's not really inside a person exactly. So I hope that kind of makes sense. Yeah, I was talking about different types of neurons. Glutamatergic neurons are neurons that you would have inside your brain, basically. Motor neurons are neurons that connect to muscle cells that fire or make your muscle cells activate. Those are the kinds of neurons that are of interest in HSP, we believe. So their company is doing studies with motor neurons now as opposed to the glutamatergic neurons, which is probably of more interest to us. She talks about autosomal dominant, autosomal recessive. Again, those alleles I was talking about earlier. Remember, you get one from each parent. So you have two alleles, two genes that express every possible trait that your body needs in every cell again. So if you have a disease that is autosomal dominant of those two genes that you get from each parent, if only one of them has a defect, has a mutation, you will express the disease, you will have the disease. If it's a recessive trait, remember you get those two genes, one from each parent. If one of them is defective, it doesn't matter. If the other one's okay, if it's not defective, if it's a recessive trait, you will not express. You will not have the disease. You will be a carrier, though. Both of them have to be defective for you to have the disease. So her company is interested in the moment in looking at possible treatments for autosomal dominant traits like SPG4, for example, because what they're looking at is, can you make the other allele, the other gene that you have that is in a dominant trait is not defective, do its thing and make that little protein that you need, and basically tell the other one, shut up. The one that's defective, to just stop doing it. If you could give somebody a drug that would do that. That's kind of what they're doing. And when she talks about up-regulating a gene, that's what she's talking about. She's talking about making it do its thing more. And it's complicated. We don't really totally understand how genes are told to turn on and off, like to actually make RNA in a particular time or not make RNA. And that's what they're trying to look at as a possible treatment for various forms of HSP. Hi, I'm H.McClaren. I'm going to try to explain a few terms in Dr. Blackstone's presentation. He talks about a synapse. The synapse is essentially the connection between one neuron, one nerve cell, and another nerve cell. My fingertips are the ends of one nerve cell and another one. There's a little tiny gap between them, and they actually output a chemical called a neurotransmitter, and that is output from one nerve cell, and then it's received by receptors on the other nerve cell, and that causes the next nerve cell to trigger along the line. It's pretty cool. I think probably everybody knows he talked about symptomatic treatment and therapeutic treatment. Symptomatic treatment just alleviates the symptoms, the spasticity, that kind of stuff. Therapeutic would be getting at the sort of the underlying cause of the disease. A couple other neuron terms he spoke about. So a neuron is, they're typically very large cells. Like some of the large motor neurons can be up to 1 meter long in your spinal cord. So very, very large cells. They have like a body and this sort of an arm, like a very long skinny part, which is called an axon. So that's what an axon is. So nerve signals sort of transmit along these. If my arm is the axon and my fingers are where the synapses are, so they kind of transmit along these long skinny parts of the nerve cells. And you've got, especially in your spinal cord, you've got in your brain, you've got a lot of nerve cells bundled together. Very important they don't interfere with each other. The signals passing through the surface of one nerve cell don't affect another one. So these axons are covered with this layer of material called myelin, and that's actually created by a different kind of cell called an oligodendrocyte. So when Dr. Blackstone talks about myelin and oligodendrocytes, they're cells that aren't nerve cells, but they're related to nerve cells and how they pass signals to one another. He spoke about endoplasmic reticulum, which often we just use the short form and call it ER for short. That is like a part inside a cell. It's basically a transport system inside the cell. It has a lot to do with protein synthesis and in nerve cells in moving materials from one end of the nerve cell to the other. He talked about various different gene therapy techniques. AAV, basically, that's actually infecting a cell with a virus. The way viruses work, they contain DNA or RNA in some cases, and they sort of attach to a specific cell, and they kind of shoot their little DNA into the cell, and they like take over the mechanism of the cell. That they cause their own the body cell to manufacture the parts of the virus. That doesn't sound like a good thing to do to a neuron. So what you do, you use an AAV as a deactivated nerve cell that's no longer, or sorry, a deactivated virus. You stick in your own gene, your own DNA that you want to input into the cell. So you basically use the virus as a vehicle or a vector, they call it, to transport your new gene that's not mutated into a cell inside a person's body. It's kind of a neat technique. ASO is basically, it's a means of deactivating a specific gene so it will no longer function. So that's turning off a gene inside the cell. CRISPR, he mentioned CRISPR, you've probably heard that term, and prime base editing. Those are methods of modifying a specific gene inside a cell, of changing it so it does something else. And I think that's all the terminology I wanted to talk about in Dr. Blackstone's presentation. So thank you very much for listening. Hi, my name's H.McClaren, and I'm going to talk a little bit about the presentation from CPATH from Tom Hart and Colin Hovinga. I'm a science guy. I'm not a regulatory person or a research clinician, so it's not my area of expertise. I do know a little bit about it, so I won't go into great depth and detail about what they were talking about. Essentially, what CPATH does is they bring people together who are going to collaborate, going to work together to develop a new drug, a treatment, do clinical trials for a drug treatment. And that, for patients, for folks who have a disease, that can be a frustrating and complex process and difficult to understand. There's a whole lot of math, believe it or not, and statistics and science involved in the process of approving a drug. And so every government in every country has a regulatory agency. In the United States, it's the FDA, the Food and Drug Administration. In Canada, it's Health Canada, who are responsible for approving a drug for use for prescriptions. And they each have certain sort of standards that they want to see in whether a drug is approved or not. There's a lot of pretty complex procedures and steps that have to be gone through, and that's essentially what Tom and Colin were talking about. What CPATH does is bring together people who are able to sort of work that path, bring them together with the regulator, make sure everybody's working together, also provide sort of statistical and calculation tools that folks can use to get that regulatory approval that they need. the proper kind of methodology that they need. That's complex. Like, in order to get a drug approved for use, you have to prove that it actually works, that it actually does what it's supposed to do. Well, what the heck is it supposed to do? Oh, help HSP. Well, how do you define that? You have to define it, and there has to be measurable outcomes. For, oh, people take that drug. These specific outcomes that we want to see that make them better are achieved. And it's not good enough. Oh, one person gets the drug and it works for them. I don't know. You see that in movies a lot. Oh, the brave scientist takes the drug that he's testing and it works, and oh, therefore it's good. That's not good enough. It takes more than one person. Like, sometimes people just get better just through random events. Um, you have to have a certain number of people in your study for it to be statistically valid. You have to have measurable outcomes. That's what ties into the talk about biomarkers, like specific things that you can measure that relate to a disease. That, well, if you take this drug, the amount of this particular substance in your blood decreases. So it's actually doing something. It's actually doing some good. That ties into the natural history that we know the typical sort of progression and stages of the disease, and then we can test and see if this drug that we're testing actually improves that. We really don't. I know it's frustrating for patients sometimes. Like, just get the thing on the market and let it do its thing. It's really, really important to ensure that a drug that's approved for use does what it's supposed to do and doesn't do bad things, because in the past that has happened where you know all these complex methodologies weren't followed for some drugs. Like, if you want to Google thalidomide, for example, you'll learn all about that. So yeah, it's important. It's complex. It's expensive in particular, and CPATH is a way forward to make all of that work better, where we're bringing various people together, researchers, maybe drug companies that want to fund the research because they're going to make money at the end supplying a drug, and the FDA in the States or whatever country you're in, the regulation agency of everybody work together, pulling together to make the research have a successful outcome. That's what this presentation is all about, and I hope that little explainer is helpful for you. Thank you. Hi, my name's H.McClaren, and I'm going to explain some of the terms that Dr. Fakhari uses in his presentation. He talks about a disease model. A disease model is an animal like a mouse that presents the disease, that has the disease, or it could be basically a cell model, like a tissue or a group of cells in a petri dish that exhibits the particular disease. Basically, it's something that you can test before you go to human trials, like to find something that's actually going to be effective. The words phenotype and genotype. Genotype means your genes, your specific series of DNA. Phenotype is your characteristics. So when you're studying a disease, it's really important to understand how the one connects to the other, how the genotype, the specific sequence of genes, is going to give you a phenotype, specific characteristics of the disease. And that kind of ties into what Dr. Fakhari talks about in natural history. That is the natural history of the disease is essentially what happens if you have that particular disease. What are the specific characteristics of it? We want to, for the purposes of research in particular, have these things really well defined. It's important because when you do research, when you're doing a trial of a treatment, a medication, you have to know, does that affect the characteristics of the disease? Does it make them go away? And then exactly how? Or does it make something else worse? You have to know, it has to be very specific. So that's why it's important to study the natural history of the disease, to show how it progresses with time as well. Um, and that's actually quite difficult in the case of HSP because it presents so differently. Um, sometimes you have childhood onset, sometimes it doesn't happen until the person is much older. There's so much variability about the thing. It's not well understood exactly why that happens. He talks about different sort of modalities or methods of possible treatments. So when you talk about a small molecule, that is essentially a drug that you could give the person that affects the disease. Um, it might be alleviate the symptoms, and there are a number of treatments for that. But ideally, we would like something that like goes to the right to the target and affects the particular mechanism that causes the disease. So there are various ways to find these small molecules that affect the disease. Protein replacement means, so proteins are like large molecules inside the cell. They're like little machines that actually do stuff that accomplish the tasks that the cell needs to do to keep it alive and to do, whatever it is that it does. In the case of neurons, sending nerve signals to muscle cells, that kind of stuff. Proteins are the machines that actually do all that. So, basically what causes a genetic disease is some protein in the cell is made wrong. It's defective. Well, if you could, squirt the protein into the cell, that would fix the problem, wouldn't it? Um, Not really sure how we go about doing that. It's complex. Um, and antibody, it talks about antibodies. That would be a drug that would disable a defective protein that would sort of bind to it and cause it to not work anymore. And why would we want to do that? Well, in some cases with HSP, um, it's not exactly clear whether the problem is that the genetic mutation causes a protein that doesn't do what it's supposed to do, or does it create a protein that does too much of what it's supposed to do, or does something totally that it's not supposed to do. So doesn't do what it's supposed to do, that's called loss of function. Does too much is called excessive or detrimental function, if it does something wrong, basically. Um, so, if we're going to, for example, use an antibody that disables the protein, well, we better know which one of those it is first. And he mentions three things I'll just lump together for now: ASO, gene replacement, and gene editing. Those are all various different ways of actually modifying your genes. Essentially, well, ASO is more modifying how the gene is expressed, how it actually makes proteins at that level. Gene replacement and gene editing are kind of self-explanatory. Gene replacement is where you shove in a new whole entire gene that makes it the proper protein. Gene editing is you modify the gene that's in the cell to make it work properly. Dr. Fakhari mentions several times about the really vastly different symptoms and the many different genetic sort of abnormalities that are causes of HSP, and that obviously makes treatment difficult. Three other terms he mentions after that are autosomal dominant, autosomal recessive, and de novo variants. So in every cell, you have 46 different little chromosomes. These are the little molecules that contain your genetic information. 23 of them come from each parent. So you have one from your mom and one from your dad. 22, well, 44 of those are called autosomes, 22 from each parent. And the last one, the 45th and the 46th, each comes from each parent, um, are the sex chromosomes. They're called X and Y. If you happen to get an X chromosome from both parents, that makes you female. If you get an X chromosome from your mother and a Y chromosome from your father, that makes you male. So males have one X and one Y. Females have two X chromosomes of their sex chromosomes, and the other 22 are these things called autosomes that are kind of more or less the same from each parent. So an autosomal trait is one that's on one of those 22 chromosomes that are not X or Y. Now, what's dominant and recessive? So you get two copies of each chromosome from each parent. Each copy does the same thing, basically. So I'll give you a simpler example. So on one of your chromosomes, there's a gene that determines your eye color. And it can essentially be brown or blue. Turns out, if you inherit the gene for brown eye color from one parent and the gene for blue eye color from the other parent, you will always have brown eyes. So the brown gene is what we call a dominant gene. Okay? To get blue eyes, you have to inherit the gene for blue eyes from your mother and the gene for blue eyes from your father. That's the only way you're ever going to get blue eyes. That's what we call a recessive gene. Some forms of HSP are dominant and some are recessive. So essentially the difference is, as far as the disease is concerned, is if it's autosomal dominant, you have to inherit it from only one parent in order to actually have the disease. If it's autosomal recessive, you have to inherit it from both parents. You have to inherit the mutated gene from both of your parents in order to have the disease, like to express the disease. And that means also you can inherit one copy of a recessive gene from one parent and not actually have the disease, but be a carrier of the disease. What's a de novo variant? De novo means new, basically. It's a fancy way to say new. That would mean you didn't really inherit that mutation. That mutation occurred in one of the cells, the sex cells, the egg or the sperm cell that made you. For the first time that happened. So that's a de novo variant, and it does happen. In terms of genetic testing, your genome is all of your genes in your cell. Your exome is just the parts of your DNA that actually make proteins, and that actually turns out it's a very small fraction of all of your DNA. It's quite complex to do genetic testing. I won't get into the details of that, but when typically, more in the past, I guess, when you got genetic testing, they measure only specific little pieces of your chromosome. They don't actually do the whole thing because it's very expensive and complex. That is not necessarily the case anymore. It's much cheaper and easier to do more genes. So one of the things they're trending towards now is doing whole exome testing, which is basically if let's say you go to a doctor and you present with symptoms of HSP and they want to get a genetic diagnosis, instead of just testing specific little pieces of genes to look for specific, standard like SPG7, SPG4, those sorts of things. They look at your whole dang exome. They measure the whole thing, all of the parts of your chromosomes that make proteins, and see if there's anything wrong with any of them. Anything different from the normal. What's a biomarker? A biomarker is some sort of way to show that the person has or has not the disease in a relatively easy way, like a blood test, as opposed to maybe genetic testing. Because even genetic testing, there's something like 30 to 40% of HSP uh, does not have an identifiable genetic um, cause still. So a biomarker would be something that, well, if you have HSP, you have this biomarker, this specific thing that we can measure. And that's very useful for clinical trials and things like that in particular, because you know for sure that that person has that specific uh, thing. He talks about screening for small molecules. So that kind of ties into the whole having a disease model, having a, well, in particular, having something like a cell line that you can produce in a petri dish. And you can essentially, using sort of modern techniques nowadays, you can just test a whole bunch of drugs and see, like if you have some identifiable thing in those cells that, oh, if that cell has HSP, it looks a certain way, and you can just essentially spray it with a whole bunch of different drugs, molecules, and see if any of them cause it to look like a regular cell. And that way, even though you don't necessarily know exactly what the mechanism is that's causing the disease, um, you can test and see if something's going to affect it. And there are a couple, as the speakers have mentioned, um, a couple or one particular small molecule that's approaching clinical trials. So that's about all I want to say about Dr. Fakhari's discussion.